So, thanks for this embarrassing introduction. I think that I'm here because Zvi Zellinger recruited me to the department, to the university, and he was an amazing scientist, advisor, and friend, and I think he definitely deserves this tribute of a conference. We miss him a lot. So, I'm going to talk about a totally different level, about non-coding RNAs and traumatic memories. But to continue the narrative of the previous lecture, I'll start from the link between trauma and liver metabolism. So that I know is not a straightforward link, but bear with me. And I would also like to push forward the theory that we developed our reactions to trauma evolutionarily, and that some of the reactions of the wild man in the desert to traumatic conditions developed because that man in the desert was running from a lion or had to hide in a cave when there is no food for the next 2 months. And we still carry the same machinery, although we are stressed due to totally different signals. So I don't need to tell you that many people turn to the fridge when they're stressed. So there is a link between metabolism and traumatic reactions. And sure enough, we work for many years on the cholinergic system and the way it affects gene expression. I'll talk about PTSD also from the perspective of the Israeli community. You know, we are in the world center of stress. And finally, I want to share with you unpublished findings on the difference between men and women in brain gene expression and traumatic reactions. So, this being a school, here is the cholinergic signaling system for you. And that probably would be the only signaling slide I have. So we are talking about a reservoir of compounds that can go out and come back in to the reservoir. It's a very clever reuse of materials and reach to the other side of the cell, to the synapse, through receptors and or get degraded by enzymes such as acetylcholinesterase. Actually, there was a festive dinner in the Hebrew University, and they set me same table with a French clinician. And he takes his BlackBerry out and says, "So how do you spell your name?" I tell him how I spell my name, and he says, "Oh, acetylcholinesterase." So that's how I spell my name. Okay. So acetylcholinesterase breaks down acetylcholine, but again, the choline transporter will bring the remnants back. So this is a very clever system. In the brain, cholinergic signals show different pathways that communicate between many different parts of our brain. So it's a very complex system altogether, and to bridge between the pathways and the components is not a small task. Now, just to make it more relevant to our daily life, there's an accumulation of papers recently on a adverse reaction of aged individuals to anticholinergic agents. And they come from the United States, from England, from Europe. Apparently, off-the-shelf drugs like anti-allergy pills may increase the risk for cognitive decline in aged individuals, which was not known. It's probably learned today because people live longer. So there is a relevance to our daily life that keeps in my mind, and we need to refer to that all the time. So of all of this, I know more, or I know a little, only about the molecular regulators of cholinergic signaling. So the cholinergic system to me seems like an onion. There is a layer to be peeled, and then you find another layer and another layer. And years ago, we cloned the cholinesterase genes in humans before the Human Genome Project, so that was a difficult task. Then we found mutations in the cholinesterase genes that increase the susceptibility of individuals to exposure to commonly used insecticides, just people that live next to orchards, not occupational exposure. And then Kevin Tracy and colleagues found the amazing fact that in the periphery, acetylcholine signals can block inflammation by changing the NF-kappa B signaling pathway. So we have relevance to those pathways that were talked about in this meeting. And we have studied microRNA controllers of cholinergic signaling, which have a tremendous advantage in that they are translationally usable. You can synthesize an inverse sequence to a microRNA, which is a small chain, and use that to treat animals or expose cells and so on. So let's talk about microRNAs. Those are very small agents, about 20 nucleotides in length. They can each target many different targets, and that makes them amazing tools. Often I think that they're so clever that if they didn't exist, someone should have invented them. But they do exist. So I went to a conference several years ago, and a colleague asked, so how do microRNA genes evolve? Which was a good question. So I said, look, they're so small that the statistical prospects of such genes to undergo evolution is very small. I would suspect that the targets get evolved to adapt themselves to microRNA regulation. And then I talked about that in our weekly lab meetings. And this student, now a postdoc seeking his way to come back to Israel, Shachar Barbash, said, but we can check that. We can get back to the genome data sets and find out in our genome, in the human genome, how long back can we date microRNA genes? That is, which microRNAs existed in our genome as early as vertebrate evolution, mammalian, or big primates. And we can ask for variation, so we can scale the changes in microRNAs and compare that to changes in their targets. So we've done that. I mean, Shachar did that, and I forgot to add that I like to follow our research in the perspective of the people who led specific projects. So what Shachar found was that in the early vertebrates, there were very few microRNAs, but they existed each in many copies and controlled very few genes. When mammals developed, we find now younger, seemingly younger microRNAs. They're less well organized. They exist in smaller number of copies, but they control more genes. And when we look at the big primates, we find young microRNAs. They are really disorganized, but they control many different genes, mainly in the brain. Well, the brain expresses more transcripts than any other tissue in any case, but this is particularly true for microRNAs. Now I'd like you to look at the scale, at the color scale. So what you see here is that the younger the microRNAs are, the lower is their variation. But when we look at the targets, it's the opposite. So the targets and the microRNAs talk to each other, and there is always an adaptation of one in view of the other. And the brain is the most important tissue where we could look for such microRNAs. Okay, so this fits a picture where microRNAs evolved to help us use tools, come to conferences, and so on and so forth. But which specific targets do they have? To do that, we set back to the cholinergic system, because today we know about 2,500 microRNAs. That's too much to focus on. And Bettina Nadop in the lab looked for, or she made the following the following statements. She said, okay, I read the papers. One microRNA often targets many proteins involved in the same pathway. We believe that cholinergic signaling is a pathway. Therefore, there should be microRNAs that control more than one protein or one mRNA involved in that pathway. And she simply screened the literature, taking cancer away. We are not cancer lab. And she found about 30 microRNAs, 1/3 of which are primate specific. So if you look at that level, we as big primates acquired another layer of regulation over cholinergic signaling. This is the molecular structure of microRNA 132 at atomic resolution, and it targets acetylcholinesterase. And we initiated our studies with that microRNA, which is conserved, so it exists also in mice. Then we progressed to microRNA 608, which is primate specific. And finally, I'll tell you about another one, 125B, which we started looking at very recently. So again, we see here the evolutionary concept we predicted. And our ERC project was entitled Cholinomers, microRNA regulators of acetylcholine signaling. Okay, so microRNA 132, they all have only numbers, they don't have names, which always reminds me of the guy who sat and read the telephone directory. And his friend says, why do you do that? And the guy said, I agree. The narrative is not that exciting, but the characters. So that's true also for microRNAs. They're really exciting. And when someone in the lab gets interested in a microRNA, they come and ask me, because I have the telephone directory in my head, sort of. Okay, so this is Morhanan, and she looked at the function of microRNA 132, which is induced under any stress in the hippocampus, for example, and she asked the question, does that affect cognition? And she found that, yes, indeed, this microRNA controls stress-induced impairments in cognitive decisions via suppression of acetylcholinesterase. And we offered this cover page, but molecular psychiatry stole this picture for their website and didn't put it on the cover page. So if you submit a paper there, it's with our picture. So that again says that under traumatic situations, there'll be blockade of esterase, therefore increase of acetylcholine signaling, elevated blockade of inflammation, which is important for the brain's reaction to traumatic situations. So how can you study that? We made a mouse. This is Yiftach Shaked, and what we have done was to overexpress acetylcholinesterase in a mouse, but take off the microRNA interacting domain. So we have a mouse with reduced cholinergic signaling, but without the impact of microRNA 132. And the question was, what would happen to that mouse? So this is what happens to that mouse. It runs. I mean, this is normal speed. It's not sped up. These mice run all day long. They're about 20% lighter than litter mates. You would say that's very good, physical activity, healthy, great. Except that Shai Shoham looked for their behavior in serial maze, and the normal mouse on top learns very quickly that there is apple juice at the other end. But the engineered mouse in the bottom keeps going back and forth, and it makes decision errors, and it gets confused. It doesn't remember where it left its keys. It doesn't know where are the glasses. And I need to be very careful with that, because once a woman in the audience said, "Oh, just like me." So I was... Anyway, so these mice are both anxious and stupid. In addition to that, they in addition to that, they have huge inflammation markers in their intestine. And of course the question is, does that reflect what happens in humans? So we collaborated, and this is Shani Shanar, who was a postdoc in the lab then, and we collaborated with clinicians who take off inflamed pieces of the intestine from Crohn patients in surgery, and they remove the entire inflamed region until they get to a quiet domain. We compare the levels of microRNA 132, and in inflamed intestinal tissues from humans, it's sky high, about an order of magnitude higher than in the quiet domains. So this is relevant also for human beings. And then came another clinician, this is Hanan Meidan, and he came to me with a study he has done to look for an overlap between anxiety and metabolism. And he found several overlapping microRNAs, including 132, so I knew he must be correct. So then I thought, what if in the old cartoon, the fat man is anxious for a reason? It's not a coincidence that the thin man is always happy and the fat man is anxious. And if that is the case, then we should look at the liver, because the liver accumulates fat, and this is the site that would reflect this evidence. So I asked Geula Hanin in the lab to create a mouse with overexpressed microRNA 132. And she failed. 74 microinjections, it didn't work. But Geula is stubborn, so she changed the promoter to a collagen promoter, which is not expressed in the brain. We followed the embryonic development, and these mice were aborted at day 14 when the brain develops. So too much stress is not good for brain development, which makes sense. And when she has done the collagen promoter, she got viable mice. And she called me and said, look at the liver. So you can see here that the liver is white, full of vacuoles, stained for fat. And I said, no, this must be an error. The transgene got into a fat-controlling genome domain. So she made 2 more lines, and they had the same phenotype. Then I was convinced. And in addition, these mice had triglycerides counts that were very high. Their bad cholesterol was very high. If they would go to their family clinician, they would be reproached. They're unwell. So now the challenge was, how can we prove if this is a causal event? And we didn't want to prove that in transgenic mouse, which are sort of artificial. So we went back instead and sent mice to McDonald's for 17 weeks. In other words, we bought a very expensive fat-rich mouse diet. When you feed mice in that, with that diet for 17 weeks, they almost double their size. They accumulate a lot of microRNA 132 in the liver. And then we could inject them with our antisense agent, which is just inverse to this microRNA, chemically protected so that it wouldn't be degraded. And thanks to the nature's rule, anything we inject intravenously goes into the liver. So we have a drug that goes directly to the liver and indeed reduces the level of the microRNA 132 almost to nothing. Okay. So this is what happened to the liver. These are photographs from Geula's mobile phone. And you see here the outcome of 1 injection, 1 treatment, and within days, up to a week, the liver shrinks back to normal size, becomes dark colored, loses the fat staining completely. And this is a reversible process. After 4 weeks, the liver starts accumulating fat again. We can re-inject and repeat the impact. So, in other words, it could be that the wild man in the desert had fat accumulation as a positive selection advantage, because these were people who could survive in periods when there was no food. And that we inherited that capacity, although, as I said, we are getting upset because of different reasons. So there is an old theory from 1962 by James Neal, who said that several of our genes are thrifty genes. They remained in our genome because they were advantageous many, many years ago and are still there. If that is the case, then are there people who suffer fat accumulation for inherited reasons? And apparently, unfortunately, there are a lot of communities like that, mainly those communities known for inbreeding, like Ashkenazi Jews. So we tend to accumulate fat because we inherited those genes. The LDL receptor, the cholesterol receptor, includes at least 1,500 mutations that are well known for such communities. So we tried to treat those mice as well. We bought them in Jackson, mice without the LDL receptor. They react also to the same antisense treatment, but via totally different target of genes. So it's a context-dependent effect. And if we look at the measured versus predicted levels of LDL and the ratio to the VLDL, we find that any transcription factor that we looked at will induce a very minute effect. But the transgene elevates fat accumulation like crazy, and the antisense agent reduces it like crazy. So here we have a microRNA that has a different position. All microRNAs are equal, but some are more equal than others. Okay, why is that? So we know how fat is produced. We know the compounds. We know the enzymes that produce these compounds. We know the transcription factors that control those enzymes. And microRNA 132 controls all of the transcription factors. So it practically controls FOXO3 and P10, which we heard about in the previous talk, and P300. It sits on top of a pyramid, and it's really a super regulator of metabolic and stress reactions. Okay, so we put mice in a metabolic cage. And what you see here is that the the antisense-treated mice change the pattern of fat oxidation, carbohydrate oxidation, and energy expenditure. And interestingly, they do that mainly at night, which parallels our day, right? Mice are nocturnal animals. And then we look in the literature, in the brain, and we find that microRNA 132 controls the circadian regulation of neuronal gene expression. So it fits beautifully. So we have a drug that switches from lipolysis to lipogenesis, and once we publish that paper, 1 week after that, we get a telephone from a company producing antisense therapeutics. They signed an agreement with us. They tested our agent. 3 months later, they call me and they say, "Lady, you don't know what you have in your hands. The data are much better than what you think because our chemistry is better." So now we are waiting to see if that will produce. Okay. The problem we had is that the changes in gene expression in terms of long transcripts were very minimal. And then we started finding papers in the literature saying that microRNAs can be degraded by their targets. So it's the opposite concept to what I started with. in the established literature, we say a microRNA stops translation and induces destruction of its target. Now we find that certain microRNAs are destroyed by their targets, and totally inverse process. So then we said, okay, we suppress microRNA 132, and that increases the expression of its targets. If some of these targets can induce its destruction, then we'll get further destruction. No wonder one injection is enough. And we might see destruction of other microRNAs. How can we test that? We inject mice either with the antisense to microRNA 132 or with an antisense to the primate-specific 608, which doesn't do anything in mice. And this is the project of Hermona Soreq in the lab, and this is what she finds. After 1 week, there's a massive change in a set of microRNAs, each of which targets another pathway in the process of lipid production. So this is unpublished, and we are still working, but it has been validated by qPCR. And we now will test what are the consequences of antisense agents towards these microRNAs. Okay. So far for the liver. And now we say the following. Everyone talks about personalized medicine, but so far personalized medicine has not really happened. How would a personalized medicine be relevant for the microRNA field? Well, it would be relevant because we kept considering only mutations that change protein production. And now we know that there are domains like the 3' untranslated region of messenger RNAs that carry binding sites to microRNAs. So if we change that, we might cause a difference, although we didn't change the protein. So Alon Simchovich in the lab looked for differences in these sites, and these are like 6 different differences in the microRNA binding site of the acetylcholinesterase transcript. One of them binds 132, and another binds 608, which, as I said, is primate specific. So what happens to people who carry such single nucleotide polymorphisms? Not mutations. The high incidence might reach half of the population, and it's by law higher than 1%. So we tried to look at that. So that's the other half of Geula's PhD. She's now, I should have said, a postdoctoral Newton Fellow in Anne Ferguson Smith's lab in Cambridge, UK. And what Geula looked at was a cohort from American volunteers, young and healthy and all doing sports. And she genotyped them and found a change which blocks microRNA 608 binding. And those volunteers also went through psychology questionnaires. And those who carried that change showed higher anxiety. Although it's just one nucleotide change in a non-coding region. They also had higher blood pressure, 34 years of average. So these are healthy individuals. Not clinical yet, but a caution. And they had much higher acetylcholinesterase levels in their blood. And then we got from the Netherlands Brain Bank tissues from carriers and non-carriers, and we found that carriers of this change express 40% too much acetylcholinesterase in their brain. A lot. Which is relevant because the only drugs given today to Alzheimer's patients worldwide in all Western countries are acetylcholinesterase blockers. And this is given based on one criterion. Anyone? Body weight. Nobody separates between men and women, and definitely no one uses personalized medicine. But 40% more, I went to my neurologist friends at Hadassah and asked, how do patients react to these drugs? Well, some of them react beautifully. We give them a pill, 15 minutes later they remember the names of their grandchildren. And others do not react at all. So there you are. If they have half more of the enzyme, why should they react? So again, we have something that might have been an advantage to the old man in the desert who needed to be more alert than we do. But perhaps it's not a very good thing today, and definitely personalized medicine should look for the implications. So Alon proceeded to look for what is the impact of this change on traumatic reactions. And we collaborated with Professor Talma Hendler from Tel Aviv University, who is a psychiatric expert, and she recruited paramedics from the Israel Defense Force, who are definitely stressed during their army studies, army experience, and we had 69 volunteers. We genotyped their DNA. 13 were carriers of this SNP. Now, what did we look for? In post-traumatic patients, we know that the region of the amygdala in the brain is hyperactivated and the prefrontal cortex is downregulated. What we did was to show these volunteers a section from Silence of the Lambs, which is really stressful. And indeed, carriers of the single nucleotide polymorphism showed elevated amygdala activity. So that was predictable, but then they showed even more elevated prefrontal activity, which blocked the amygdala. In that case, they didn't suffer any traumatized symptoms, as tested by psychiatric experts. So for me, that gave two messages. First is, it's good to know we all carry some DNA differences, and some of them are probably not favorable. So that our brain can overcome that is very good. Also, hyperactivating the prefrontal brain can overcome stressful experiences. And indeed, we live in a stressful era. So it's good to know that you can train to overcome that. Again, here's one more microRNA, which seems to be more equal than others, sit on the top of a pyramid. So then we moved on to really traumatized people. There are in Israel today 301 ex-war prisoners from Syria and Egypt who came back after the 73 war. We belong to a center of excellence called ICOR, which combines researchers from different universities, clinicians and basic scientists, to look for the impacts of mass trauma. And we recruited about 100 of these people, and Nimrod Madrer in the lab is looking at their gene expression and genotype, and what you see here is that 74 of them came back with PTSD, not surprisingly. The amazing thing is that 24 came back apparently healthy, and they are being tested every 3 years, and they are fine for many, many years. The PTSD patients had 17% carriers of this SNP, which is exactly the average in the Israeli population. I forgot to say that in the African tribes, we find 34% of carriers. In Caucasian non-Jews, we find 4%. In Israel, Muslims or Jewish, we all carry 17%. So if you ever doubted the theory of population migration, here is a proof. Okay, so the PTSD patient had the same incidence of mutation as the entire population. The ones who came in healthy had double. So my colleagues in the lab say, but this is a very small number. Okay, so what would the editor say? Go back and wait for another war? I mean, this is a unique cohort. So instead, what we've done was to look for additional relevant findings. And we found a report from China, where investigators looked for single nucleotide polymorphisms in microRNAs that would differentiate between people who had head injury and died because of sepsis or not. And they found just one single nucleotide polymorphism that was relevant in the microRNA 608 itself. And the carriers of that had significantly less inflammation. So if you change microRNA 608, which probably induces a structural change, you modify a lot of gene expression. Now, how can we address that in human beings while keeping research criteria? So what we've done was to approach the NIH GTEx project, which includes a lot of individuals' DNA and gene expression profiles from different brain regions, different tissues, and including brain regions. And we wrote them about our study, our question. They approved our use of that data set, and we found the carriers of the SNPs, one in the acetylcholinesterase and another SNP in the microRNA targeting it. And these are the incidence values, and then we moved on to the gene expression data sets. And the amazing finding was that the acetylcholinesterase-induced differential expression affected the frontal cortex, but the amygdala was affected inversely. So in one case, we affect the frontal cortex, in the other, the amygdala. We have lists of genes that are modified in their expression. In the frontal cortex, the FOS and JUN genes are affected. These are known to be essential for long-term memory. In the amygdala, we had the shunt gene, which I'll show you in a minute. The fold enrichment values were significant for cytokines and myelination in the frontal cortex, and for translational initiation and protein targeting in the amygdala. And these are the shunt genes, which I was very happy to learn are crucial for differentiating autistic brain from normal, and are apparently modified also in a lot of neuro system diseases. So that brings me to the last topic in this talk, where I looked for studies on nervous system diseases, mental diseases. There are two major ones, schizophrenia and bipolar disorder. There are two science articles from this year saying that they both belong to the same spectrum. So don't ever regard schizophrenia as distinct from bipolar, but think about them as a continuum. Just like autism, it's a spectrum. And I looked at that study, and frankly, I was upset, because we know from a lot of papers that males and females react differently, both to bipolar and schizophrenia. The authors looked for similarities. Nobody thought about differentiating men from women. So again, this is a personalized medicine aspect that is really important. When I tell colleagues, you know, that all of the drugs we use were only tested in males, they don't believe me, but this is a fact. So we decided to look for differences. And this is Sebastian Lobentanzer, who looked for data sets from 139 schizophrenic patients and 82 bipolars, differentiated them to males and females. And I don't want you to read the text, but note that the blue and the pink are very different. There are huge differences that are highly statistically significant in gene expression in the brain of patients with schizophrenia and bipolar. And in the schizophrenic patients, we found that one of the most significant changes was response to organophosphates. That's my turn again, because organophosphates block acetylcholinesterase. So here we come again. So I said, okay, let's focus on cholinergic signaling, which will enable us to look at really what happens in these brains. So we found single nucleotide and single cell polymorphism data sets, which I believe you'll hear about tomorrow by Nomi Chaviv, or at least part of that. And we looked for the differences in the cortical tissues from where those data sets we studied came. The amazing difference was we didn't find nerve growth factor impact. Now, cholinergic neurons in the deep brain nuclei are known to react to nerve growth factor. This is what was known for decades in the field, and that was the belief. Apparently, in the cortex, it's very different. And that raised the question, so can cholinergic neurons in the brain react to block inflammation? Because the other pathway that was really affected in schizophrenic and bipolar brains was inflammation. Now, one thing that is important, we could find that those genes are targeted by conserved microRNAs, a big part of which are primate-specific. There are transcription factors that are selective, and we could look for their segregation both between men and women and between schizophrenia and bipolar. Here is what we find. The cholinergic nerve cells in the human cortex apparently react to a leukemia-inducible factor, which we heard about 2 days ago. So they do not respond to nerve growth factor, rather they react to interleukin-6, LEAF, or CNTF, and they would use the GP130 receptor to induce activation of clock genes. Cholinergic signaling is notably known for its circadian regulation, for microRNA control, which included a lot of our friends, the numbers of which we knew, and for acetylcholinesterase and the receptor genes, as well as transcription factors that were found in nematodes to induce development of cholinergic neurons. So we have a hypothetical biochemical process. So Esther Bennett in the lab handled as an experiment that where we sequenced the outcome of CNTF application on a neuronal cell line of human female origin. And what happened was these cells developed cholinergic features, and they changed the expression of transcription factors, some of which are primate-specific, of target genes, many of which are known from schizophrenia and bipolar literature, and which affect a lot of processes that we found familiar. If you take those targets and go back to the original data sets we have, you find the schizophrenic-associated transcripts, the bipolar ones, and ones that are, the purple ones, are in between, are relevant for both diseases. Many of these are female-specific, not surprisingly, because the cell line is female-originated. But again, we would say here that it shows the great distinction between males and females. Last but not least, we took the top microRNA and we proved that indeed it targets acetylcholinesterase by the classical viral tests. So here we have a third microRNA which is conserved and sits on top of a pyramid and affects the capacity of nerve cells to react as cholinergic. And most of the genes we find are indeed circadially controlled. So we do have a full pathway leading from existing data sets that were looked for similarities, but actually show a lot of differences. So how do we go about it from now on? I'd like to end with the model developed by Nadav Yayon in the lab, who developed an algorithm that enables us to look for cholinergic neurons structurally by an... Oh, I'm sorry, something is wrong here. It's thinking. Okay, let's let it think. We have mice where all the cholinergic neurons are labeled fluorescently, and the algorithm developed by Nadav can... okay, bravo. Huh? No. Okay, we missed that. Can show us automatically the structure in the cortex, and especially in the barrel cortex that controls those features that Dudi was speaking about in reaction to the environment. I think maybe it can work when you look at it here. Okay. No, that one doesn't. Okay. Whatever. No. No, it stopped it. Oh, shit. Oh, okay. Bravo. Okay, so I came to tell you 3 different stories going from the liver to the brain and to nervous system disease. The thanks are due to the people who did the work, to the patients who donated data and tissues, to our collaborators, to the funding agencies which make this possible. And we talked about microRNAs, about personalized medicine, men and women differences. And I'd like to end by citing Charlotte Brontë, who said that the novelists should never allow themselves to weary of the study of real life. And I believe that that refers to us as scientists as well. And thank you for listening.